Bienvenue à cette conférence scientifique organisée par l'IURDPM, l'Institut de réadaptation sur la réadaptation, l'Institut universitaire sur la réadaptation physique de Montréal, en collaboration avec le CRIR. Alors aujourd'hui, nous avons l'immense plaisir d'accueillir Jason Neva, professeur Jason Neva, qui est un professeur maintenant associé, agrégé. Félicitations.
Merci,
le 1er juin. Qui vient donc agrégé à l'École de kinésiologie et des sciences de l'activité physique de l'Université de Montréal. Il est aussi chercheur au CRIUGM, donc le Centre de recherche universitaire de gériatrie de Montréal, aussi qu'au CIRCA, c'est bien ça?
Oui.
Et au CIRCA, c'est le Centre interdisciplinaire de recherche sur le cerveau et l'apprentissage. La conférence sera en anglais et je vais faire une courte biographie en anglais aussi pour Jason. So, Professor Neva completed his master's at York University in kinesiology and health sciences, focusing on behavioral aspects of motor learning. He also completed his PhD at the University of Waterloo in kinesiology, and he concentrated on neuroplasticity mechanisms as measured and induced by transcranial magnetic stimulation. And he finally completed his postdoctoral fellowship at the University of British Columbia in the Department of Physical Therapy with Laura Boyd and Jenny Stang. Is that correct?
Laura Boyd, mainly.
Boyd.
Yeah.
Okay.
Yeah.
And he focused on neurorehabilitation in clinical population, so stroke and Parkinson's disease. So today, Professor Neva will share with us his innovative work, and his presentation is entitled Understanding and Enhancing the Neural Mechanisms Supporting Motor Learning, Applications to Motor Rehab. Jason, thank you for being with us today.
Thank you so much. Thank you very much. Thanks for that introduction. Thank you, everyone, for coming today. We've planned to have this visit, meeting, and talk for a little while, so it's wonderful to be here. Thank you very much. The little update to the title is Harnessing Neuroplasticity to Enhance Motor Learning Applications to Rehabilitation, but it's essentially the same idea. So yeah, even though I'm giving my presentation in English, I'm very happy to speak in French or to take questions in English or French, and I will certainly do my best. Okay, so I'll start with Dorothy gave a very nice resume, I guess, of my research, but I'll just kind of repeat it briefly here. The focus of my research can be summed up, I would say, in that sentence at the top of the slide: investigating the neuroplasticity mechanisms supporting motor learning. And really the focus is on neuroplasticity, and that is, of course, the ability of the brain to form and reorganize itself as the underlying neural mechanism to support motor learning in healthy people or the healthy brain, and the translation of that to relearning motor function after stroke, for example. So this is what drives my research. I added this slide too, just to, as Dorothy mentioned, my lab is at the CRIUGM, as Dorothy mentioned, and my research streams, I have three main research streams, two that are fundamental and one that's more applied. The first research stream is understanding the underlying neural mechanisms supporting motor learning and motor control, understanding how we can use adjunct interventions like acute bouts of aerobic exercise and repetitive transcranial magnetic stimulation to prime the brain for enhanced neuroplasticity or enhanced motor learning, and then how we can apply all of that knowledge in my third research theme or stream in healthy aging, to age well, in Parkinson's disease, and also in stroke, focusing on how to either maintain motor function of the upper limbs particularly, or to rehabilitate or recover function of the upper limbs as well. And as you'll see with my presentation, I'll be focusing on one tool that I use quite a bit, which is transcranial magnetic stimulation, to both measure and to modulate brain excitability, okay, across all the research streams. Okay, so today for my talk, I thought that I would focus on the two parts of two research streams that I mentioned before, the second and the third, actually. So focusing mainly on adjunct interventions in order to boost the brain and behavior. For example, using acute bouts of aerobic exercise and repetitive transcranial magnetic stimulation. And I'll describe both of these, what I mean by both of them later, separately, maybe one day together too, in order to enhance neuroplasticity and in order to enhance motor learning or motor relearning function after stroke. And that's the application. So today for the talk, I'll be focusing on kind of three sections. the use of aerobic exercise to enhance motor learning and neuroplasticity. And these are mostly in healthy individuals too, and for the first section. The second section is the use of transcranial magnetic stimulation, or TMS, in order to measure and modulate the brain in people with stroke and healthy individuals too, to understand what's going on in the brain. And then the last section is some clinical application potentials and some future work in stroke, a new and exciting project that we have that's starting this summer. So the first section on the use of aerobic exercise in order to prime and enhance motor learning. So we all know that aerobic exercise, and by the way, I'm focusing on aerobic exercise rather than other types of exercise, which are also good, but aerobic exercise, we all know to a certain degree that it's good for us. It's good for our body, it's good for our muscles, our bones, our immune system, our cardiovascular system, our heart. But recent research, say in the last few decades, have shown us that there's a profound impact also on the brain, perhaps not surprisingly. And in particular, acute aerobic exercise seems to be able to, or aerobic exercise, excuse me, seems to be able to enhance neuroplasticity. Again, the ability of the brain to change and form and reorganize itself and its physiological function particularly. Other research has shown recently that exercise can also enhance motor learning, and really, really remarkably so. Even just a single bout of acute, what I'm calling acute aerobic exercise, can enhance neuroplasticity and enhance motor learning. So there's been a lot of research that's been gathering showing that, for example, an acute bout of lower limb cycling exercise, that's about 20 minutes, maybe 20, 25, maybe 30 minutes, at an intensity of moderate to vigorous, maybe even high intensity. When we perform it before or even after we practice a motor task, we can enhance the motor learning of that task. We can enhance the acquisition of that motor skill, and we can enhance the learning as tested at retention tests, a few hours later, a day later, 7 days later, even a month later. Some, in my opinion, slightly unanswered questions are, well, what's the exact effect of exercise intensity on motor learning? You know, does it need to be high? Can we get the benefit from low or light intensity exercise? I'll be sharing some work investigating that. And how about exercise type? I mentioned acute bouts of lower limb cycling exercise. And what I mean by type in this instance is, well, we have two different modes of muscle contraction. I'll talk about that a little bit more later, but we have concentric contraction. Imagine when we're walking up the stairs, our muscles are shortening when we contract. And then when we walk down the stairs, our muscles are lengthening, going against gravity while we produce force. That's called eccentric contraction. So what's the effective exercise contraction type? I'll talk about some work with that too. So jumping right into it, some work by my PhD student, Nesrine Nahom, has looked into or asked this specific question: is there a dose response effect of acute exercise intensity? There seems to be work that I meant to mention before, that there seems to be an effect of exercise intensity. There's evidence for moderate intensity to enhance motor learning. There's evidence for high intensity. But they're not all agreeing necessarily with each other, and there's not a single study that investigated all, a spectrum of exercise intensities within the same study. So within this study, Nesrine had 80 individuals, 80 young adults in a between subject design. And what we had is we had people come into the lab. They, of course, did an incremental exercise test to determine exercise intensity. And what we had is we slotted them into one of 4 groups where we had rest or light intensity exercise, moderate intensity, and high intensity exercise. So 4 different groups, right? No exercise control with the rest, and then the 3 different intensities. And the one thing that we did that a lot of the research hasn't quite done yet was control the structure of the exercise. So notice how it's all intensity interval training exercise. A lot of the previous research has compared continuous or steady state exercise to interval exercise. So we wanted to control that, the type or the structure of exercise, and the duration. And what we had people do is we had people perform each of these exercise intensities, then immediately afterwards perform this what's called a visuomotor rotation task, and it's a motor learning task. And what it is here's just a little representation of it. They're reaching to, with a modified mouse just on a tabletop, reaching to targets. But the trick of the task is that when you're actually reaching forward, for example, to 12 o'clock, the cursor is actually veering off to 45 degrees, for example, to the right, or to the left, rather. Okay, so then people have to, and people don't see their hands, so they have to adjust and adapt in order to be able to reach to approximately 2 o'clock in order to make the cursor go to 12 o'clock. So that's the task, and it's specifically a motor adaptation task. We had people practice that task immediately after either exercise intensity or rest, and then they came back 24 hours later for a retention test and no exercise retention test. So, is there an effect of exercise intensity, or dose response effect? Well, indeed, she found that yes, there is a dose response effect of exercise, acute exercise intensity. And just to orient you to these graphs, this skill acquisition, this is the performance at that visual motor rotation task immediately after exercise or rest. And then here, the retention test is 24 hours later. Okay, so this is the real test of motor learning, and this is what we're calling skill acquisition, or we can call it adaptation too. On the y-axis, we have movement time, so lower values indicate better performance, right? And we have HIIT, or high intensity interval training exercise, in red, moderate in orange, green is light, and gray is rest, so no exercise. And what we can see is that if we just look at the graph, and oh, by the way, we have blocks of practice, right? So the trials of blocks are 8 trials each. And you can see over all of the blocks of trials, you can see that everybody is learning and adapting to the task, but there seems to be a dose response effect where high intensity looks like to be the clear winner, but then moderate intensity is improving too, and light intensity is improving as well compared to rest. That's immediately after. Then 24 hours later, we can see that it's really the case, certainly for moderate and high intensity exercise. And this graph here just shows the same data, actually, skill acquisition immediately after exercise and the retention test. And this is all the individual data, but just a average of the performance over the whole session, right? So we can see kind of clearly, more clearly, I would say, this dose response effect. All of the groups are different from each other, with HIIT being the best, right? And we have that same effect here at the retention test. So there is a dose response effect of exercise intensity. I was thinking to myself that it would be moderate and high together. They would have the same effect, but it's high intensity who's the winner in this case. All right. Doesn't want to go ahead to the next one. Oh, there. Sorry about that. Oh, there. Okay, so, well, I said that part. All right, the greater the exercise intensity, the greater the motor learning enhancement. This is what we found here. An important point to keep in mind is that we have sort of an important psychophysiological effect as well to certain measures. I apologize for the how small the actual text is, but I just wanted to emphasize here too that there's also a dose response effect of these psychophysiological measures like the perception of effort, muscle pain, and negative affect. Essentially, with increasing exercise intensity, there's greater perceived effort, there's greater muscle pain, and there's more negative affect as well. So just something to keep in mind. Along with the benefits, there's these other things to keep in mind as well. Okay, so our second question, well, another question in the lab that we wanted to ask was this idea of, okay, is there an effect of acute exercise muscle contraction type, right? And Lael Youssef, my other PhD student, investigated this question where we are comparing concentric cycling exercise to eccentric cycling exercise. So like I mentioned before, concentric muscle contraction is, for example, when we are doing a biceps contraction like this, a biceps curl, I should say, where we are producing force with the muscle shortening. Whereas if I'm holding a weight that's so heavy and it's going down, but I'm resisting it on the way down, that's the muscle is lengthening as I'm producing the force. That's an eccentric contraction. The easier example is walking up the stairs, like I said, is concentric for our quads and our legs in general. And then descending down the stairs is an eccentric contraction. What we have in the School of Kinesiology, we have some And in our lab specifically, we have some specialized cycle ergometers where we can do eccentric cycling specifically. So it's a recumbent bike where we have, it's the exact same one for our concentric cycling and eccentric, where essentially the motor is pushing the pedals backwards and the individuals have to resist it on the way back. So it's an eccentric contraction the whole time. And they have to do that at a certain RPM. Okay. So that, yeah, we had a rest group, we had a concentric exercise group, classic, and an eccentric exercise group. Immediately afterwards, they practiced this different type of task. It's a motor sequence learning task, continuous tracking task, but that's not so important. But the slightly important or interesting thing is that, yeah, unbeknownst to the participants, there was this repeated sequence in the middle that was happening across every single trial with flanking random sequences. So the random sequences they could not learn, and the repeated sequence was the same all the time, so they could learn it. So this was the sequence motor learning part. Okay, and like I said, they weren't aware of that. People came back, by the way, 48 hours later after the exercise. So they did the exercise, practiced the task, came back 48 hours later for the retention test. And indeed, we did find an effect of acute exercise type or muscle contraction type, where we did find this. So here we go on the y-axis, we have RMSC, that's just root mean squared error. So values that are lower indicate better performance. That means they're tracking the target better in that case. And we have before exercise, the exercise happens, then we have the acquisition trials, and then 48 hours later, we have the retention test. We have eccentric in purple, concentric in green, rest in gray. And what we see is this immediate enhancement in performance for both eccentric and concentric exercise that actually continues for eccentric. At the last few blocks, we can see that there's a continued improvement for eccentric exercise, which was very interesting to us. And this was in line with our hypothesis, actually. Then 48 hours later for the retention test, that improvement was maintained. So the eccentric exercise group maintained this enhanced performance, and concentric exercise was enhanced more than rest as well. So essentially, eccentric exercise enhances motor skill acquisition and learning to a greater extent than concentric exercise. But concentric exercise still enhances motor learning too, compared to rest. And this is just data showing the exact same thing here, but just the overall performance, right, where we have the differences between the exercise types at acquisition and at the retention test. Same conclusion. Okay, something else to keep in mind is, along with that, is that that's actually very interesting too, the effect of exercise type on psychophysiological measures. Confirming previous work, we've shown that eccentric exercise has a decreased heart rate response at the exact same force that they're actually resisting doing the actual eccentric exercise and concentric exercise. And that's shown here. Purple is eccentric exercise, green is concentric, and gray is rest. There's increased positive affect for eccentric, so they feel better. There's decreased perception of effort, shown here, if with eccentric compared to concentric. And then during the exercise, there's decreased muscle pain as well, during the exercise. But then we did muscle pain measures because we knew that this would be the case, or thought that this would be the case. At prolonged tests of 24 hours, 48 hours, 72, and 96, we see that there's significantly increased muscle pain while we're doing standing tests, walking tests, and ascending up the stairs tests. We see this, yeah, increased effect that you know, mostly peaks at 48 hours, then it comes down, but it's still at 72 hours, there's still significantly greater muscle pain. Okay, so yeah, there we go. The original project, the original grant that we had for this, myself and Benjamin Pajot, was, you know, eccentric exercise enhances motor learning. Is the benefit worth the pain? Well, it's something that we have to consider. Is it? I can't answer that question yet, but it comes along with more prolonged pain afterwards. But there's an enhancement to motor learning. So I believe that these that have contributed at least a little bit to answering these questions a little bit. With increased exercise intensity, we have increased motor learning. exercise type, eccentric exercise enhances motor learning even more than concentric or the traditional form. Important consideration, the psychophysiological responses though, too. Okay. Pain, perception of effort, heart rate response, affect, all very, very important, particularly in clinical contexts. Okay. So now on to the neuroplasticity part, aerobic exercise enhancing neuroplasticity. And so how I measure neuroplasticity, as I mentioned before, is through the use of transcranial magnetic stimulation. This slide always takes a while to actually load for whatever reason. There we go, video. Can I play the video? Oh, the sound is somewhere. But yeah. Oh yeah, there it goes. But anyway, so transcranial, I'll just play the video again and again while I'm talking. So transcranial magnetic stimulation, I know a number of you, or perhaps all of you, are familiar with it, but I'll just briefly describe it. So transcranial magnetic stimulation is a way that we can non-invasively measure, both measure and modulate the excitability of the brain, right? So the way that TMS works is when we're using this small coil, TMS coil, and when it's engaged, it produces an electromagnetic field, and that crosses the skull, that's the transcranial part, and then it reconverts back into electrical activity that actually activate the underlying cortical tissue. And when we stimulate over the motor cortex, for example, we create descending volleys of activity that activate these corticospinal output neurons that crosses over to the other side of the body, and from the target muscles, we can see a muscle contraction, an involuntary muscle contraction. We pick that up with service electromyography, and that involuntary muscle contraction, or evoked potential, I should say, shows up as a clear waveform as the motor evoked potential. And what's really great about this is this is our way that we can assess brain excitability, and we can use this as a marker of neuroplasticity in the sense that if we do these measurements of excitability before and after an intervention like exercise, motor practice, TMS, coffee, sleep, then we can get a sense of having kind of a marker of neuroplasticity or short-term rapid neuroplasticity. All right. So using TMS to investigate these questions, we wanted to understand what's going on in the brain. We saw after an acute bout of exercise, we see these benefits to motor learning. Well, what's going on in the brain to support that perhaps? So, by the way, this measure here is a measure of corticospinal excitability, right? Because it involves the cortex, and of course, it involves the spine, and then of course the periphery too. So this is a overall measure of corticospinal excitability. We thought that it would change, right? That there would be this increased corticospinal excitability after exercise in the non-exercised upper limb muscles. But what we found was that cortical excitability does not change. We showed this in a number of different studies, and other people found this too. There's a slight nuance and some newer results to that now, but I'll tell you about that in a minute. Generally speaking, though, those exact measures that you saw in the video and that I was just describing from the non-exercised upper limb don't change after, yeah? Want me to stop? Oh, it's not following me. Is this how you do it? Yeah. How about now? Yeah? Cool. Okay. Je continue? Okay, super. All right, so yeah, basically these are just measures in white and then immediately and post 20 minutes after exercise. Values that are higher indicate greater excitability. Point is that we and others have found over and over again that there's not a change in this corticospinal excitability. But what does change is a measure of motor cortex inhibition. So we can do this using paired pulse transcranial magnetic stimulation, where we use 2 pulses in really rapid succession, like 2 milliseconds apart. And what we do is essentially we compare a single pulse to that paired pulse at 2 milliseconds apart, and we get a smaller motor evoked potential. And that gives us this idea of inhibition that's going on within the cortex. We do these measures before and after exercise. And what we found, and I've found this in several other studies, and other people in Australia and all over the world have found this as well, but there's this lessened inhibition. Values higher mean less inhibition, values lower in this case mean more inhibition, and we have significantly less inhibition after exercise. That seems to happen at a little bit of a delay too. So This is just kind of like a sampling of the effects. There's lots of other effects that are happening in the brain between the cerebellum and the motor cortex, the somatosensory cortex and the motor cortex, between the two sides of the brain too. But this just gives you a little sense of the effects. Not everybody has found these effects though. The one that I just mentioned before, corticospinal excitability, overall output excitability, nor this inhibition measure. So we decided to do a systematic review and meta-analysis of the literature, asking these two specific questions. What TMS measure is most consistently impacted by acute aerobic exercise? And what is the effect of exercise intensity? That's a big question that everybody has. Just by the way, the exercise intensity that I spoke of in the last two studies were both moderate, continuous, steady state exercise. All right, so we published this recently in Neuroscience and Biobehavioral Reviews. We had 23 studies that came from our systematic review, and lo and behold, what we found was in line with our hypothesis that indeed short interval intercortical inhibition, or just motor cortex inhibition, was robustly and most consistently decreased after exercise. That's the TMS measure that came out. Okay, and what all of this shows is all the studies are showing on average that there is this significant effect, a significant decrease in inhibition. Yeah, overall effect. And what was primarily driving that was the high intensity study, the studies that employed high intensity, and the studies that employed moderate intensity too. There's not enough data with low or light intensity yet. And then corticospinal excitability. I said there's going to be no effect. This is what we found over and over and over again, right? But I mainly used moderate intensity, moderate continuous. What we found is the overall effect was no significant effect, just barely not. But we found that high intensity, particularly high intensity interval, increased corticospinal excitability. So all of the studies showing that, and all the studies in line with what I thought, moderate intensity didn't increase corticospinal excitability. So there's an intensity factor there. So what's the most consistent effect? Motor cortex inhibitions decreased. Moderate intensity to high intensity drives the effect. But even though we have a sense of the effect of exercise intensity, to me, it still wasn't entirely clear because all the studies mostly used high intensity intervals. Sometimes they used high intensity continuous. Sometimes they used incremental test, at varying different durations, varying different intensities. So we wanted to nail down and understand this effect of exercise intensity. Asking the question, is there a dose response effect of exercise intensity on neuroplasticity as measured by TMS? So Nesrine, it's like almost exactly the same study design as the motor learning study of hers, except in this case, it's a within subject design. We had 30 young healthy participants. We did our TMS measures before and after either rest, lit, light intensity, MIT, HIT, and rest. Okay, and the hypothesis was that for me, it was going to be moderate intensity to high intensity. We're going to probably give the effect, particularly for inhibition. But corticospinal excitability, maybe HIT, but I really thought there wasn't going to be an effect. But so what we found for corticospinal excitability, that HIT showed the greatest effect. High intensity increased corticospinal excitability to the greatest extent. This is MEP amplitude. Higher values just indicate greater corticospinal excitability. HIT's in red. Okay, we have moderate intensity that actually increases immediately, but then goes back down. So HIT has this sustained effect. This is all just the individual data. I know it's small, but just to give you a sense of how everyone was behaving. But on the other hand, for cortical inhibition, that other measure that we're talking about, SICI, moderate intensity seems to show the greatest effect, where there's an increase and then it's maintained, whereas HIT tends to go down and it's not significantly released or less inhibition compared to rest. So we have this nuanced kind of dose response effect happening. So then we have a great, we had the effect of eccentric exercise enhancing motor learning to a greater extent than concentric or traditional cycling exercise. We wanted to understand what was going on in the brain from that. Okay, and the hypothesis was that, of course, it's likely that there was less inhibition or greater corticospinal excitability due to eccentric exercise compared to concentric. So this was the exact study design. It was a within subject design. We did TMS measures before and after, actually 2 time points after. We had concentric cycling exercise, eccentric cycling exercise, and rest. We did our measures of corticospinal excitability and motor cortex inhibition. And what we found was that essentially there was the same effect for eccentric and concentric exercise. They both increased corticospinal excitability. That's what this graph on the left is showing us. And they both decreased motor cortex inhibition to the same extent, both more than rest. Not exactly what we hypothesized, but still interesting. It's really, yeah. So how does exercise impact the brain? Decrease motor cortex inhibition, increase motor cortex output excitability. These measures, why are they important? Well, they're very, very important, or the markers of rapid neuroplasticity, or changes in the brain that are happening. These types of measures are very, very important telltale signs of to support early stages of motor learning. And they're very, very important for recovery of function after stroke. For example, corticospinal excitability. If an individual has a corticospinal output after stroke, there's a greater chance for recovery. If they don't, then there's a decreased chance for recovery. And also after stroke, excessive inhibition can be a negative thing for motor function as well. Important factors to consider: aerobic exercise intensity and muscle contraction type during exercise. Okay. All right, I'm going to try to go a little bit faster because I think I prepared too much. But yeah, so this next section on TMS, to measure and modulate the brain, I'm going to start with stroke because it really frames this investigation, even though there's a lot of investigation in young healthy people as well. Well, not a lot. There's just 2 main studies that I'll be talking about. So we're all familiar with stroke, so I don't need to really cover it too much. But essentially, the particular interest that I have in it is, of course, the upper limb function, upper limb paretic function after stroke, and being able to find ways and adjunct interventions like transcranial magnetic stimulation to improve or to enhance kind of motor recovery or motor relearning after stroke. And importantly, approximately 80–85% of Canadians live with persistent impairments into the chronic stage, that is 6 months to a year after stroke. Acute care has really, really, really improved. You know, a lot of people are surviving after a stroke, but a lot of people are living with these persistent impairments, particularly, I would say, from my perspective, of the upper limb too. Of course, the lower limb as well too, but yeah. So a lot of the research, at least the clinically related neuromodulation research, has been motivated by this idea of the interhemispheric competition model. Right, so what that is basically is that after a stroke, this is my horrible little circle that represents a stroke in the cortex, or in the sensory motor cortex. What happens is we have decreased motor function on the opposite side, or the contralesional side. Generally speaking, we have decreased cortical activity or cortical excitability. But then the big thing about the interhemispheric competition model is that the opposite side, or the non-damaged side or unaffected side, contralesional side, has increased cortical activity or maintained the same as before, and increased interhemispheric inhibition that's acting on the stroke affected side. So all of these things combined, the interhemispheric competition model or hypothesis says that all contributes to poorer motor function after stroke. So that's the idea. So from that, counteracting the interhemispheric competition, what do? We can use something like repetitive transcranial magnetic stimulation in order to inhibit or suppress that overactive contralesional hemisphere, to decrease that amount of ongoing inhibition to the stroke affected side, thereby increasing cortical excitability or activity, and hopefully increasing motor function. This is the idea of trying to counteract, and this is what's been done in the research quite a bit, including work that I've done actually. So just very briefly, in my postdoc, we did this study where essentially we did pre and post measures of motor skill and motor function, clinical motor function. We had 5 sessions of that exact inhibitory repetitive TMS to the contralesional motor cortex, followed by motor practice. The practice task is very simple. It's just they grabbed onto a robot handle and they were reaching to targets in a particular sequence. Okay, so it's a sequence motor learning study. So yeah, that was over 5 sessions, 5 separate days. We did that. Really hopeful that this would cause some improvement, but inhibitory RTMS to the contralesional motor cortex showed no improvement. Okay, so for our Wolf motor function test, the clinical measure of motor function, we saw a general improvement, but no additional improvement due to the repetitive TMS or the suppression of the contralesional cortex, and no additional effect to motor learning either. Okay, everybody learned the task, everybody did well, but there wasn't any additional effect. So what I took from this, and from other research that has come out too, but certainly there's a lot of evidence on both sides. But the contralesional motor cortex suppression may not always be the best solution. Okay, it's not necessarily a one-size-fits-all approach. There's meta-analyses that show that. There's meta-analyses that show that there's an effect too. But then my take on this is that outside of the motor cortex, areas like executive function, planning, somatosensory cortical regions can possibly compensate for loss of motor function after stroke, and come online and help the damaged motor cortex after stroke. And I'm particularly interested in that in terms of the interhemispheric communication. So how can we measure this interhemispheric connectivity between the two cortices? Well, one way is through a method of single pulse TMS called transcallosal inhibition, where we use single pulse of TMS, for example, to the one side of the brain, while at the same time, people are holding a contraction on the same side, right? Then the signal is sent to the opposite side and suppresses that ongoing activity, and we get what's called an ipsilateral silent period, okay, that's involuntary. So that's a way that we can measure the interhemispheric connectivity, and particularly its interhemispheric inhibition. All right, so one line of work that I'm interested in is measuring this type of transcallosal inhibition, not just from the motor cortex, because that's been done a lot in stroke, but measuring it as elicited from areas like the dorsolateral prefrontal cortex, the premotor cortex, and the somatosensory and parietal cortices as well. So what I found was that this ipsilateral silent period can be elicited with stimulation over each of these regions in people who've had a stroke. And in particular, it's very important. I'll show some data that shows that eliciting that from the contralesional cortex is very, very interesting and important. And particularly, the contralesional dorsal premotor cortex came out as one that was really important and strong, actually, in its connection to the opposite motor stroke-affected side. And here's the data that I'll just emphasize really, really briefly. Essentially, what this shows, actually, and this is actually against or opposite to the interhemispheric competition model, where essentially values lower on each of these graphs, PMC is premotor cortex. Lower values mean more inhibition. So the more inhibition means is associated with less motor impairment, and more inhibition is associated with better motor function, as measured with the Wolf motor function test, and increased grip strength too. Okay, so premotor cortex likely plays an important role for recovery of function after stroke. That's my take. So just taking a brief step back here, I'll try to go quickly through this. But my PhD student, Elnaz, she wanted to just understand, how does the premotor cortices talk to the opposite motor cortex as sort of a model for stroke recovery, right? So there's these two distinct dorsal and ventral premotor cortices. They have their own functions and so on, and they have distinct communication with the opposite M1 too. There's more inhibition coming from PMD generally in humans, and a mix of inhibition and facilitation from the ventral premotor cortex. So her objective was to investigate how these two regions contribute or talk to or influence contralateral M1 excitability. Okay, and how this can be very relevant for stroke. And specifically, she wanted to examine the different neurophysiological pathways of communication. And how we can do that is using dual site transcranial magnetic stimulation, very similar to the transcallosal inhibition measure that I said to you before, but we compare a single pulse of TMS to a conditioned pulse on the opposite side that sends an inhibitory signal, and we get a smaller MEP, and that gives us, we take a ratio of those motor evoked potentials and we get a percentage of inhibition. So what LNAS wanted to investigate essentially was a comparison of the ipsilateral silent period and the dual site transcranial magnetic stimulation, comparing premotor ventral and premotor dorsal and its communication to the opposite motor cortex. Okay, and our different pathways were trying to understand subcortical pathways and callosal pathways too. Essentially we found what I found previously, that there is a significant inhibition from the dorsal premotor cortex and the premotor ventral cortex. That's essentially what all of this is showing. But the interesting stuff, I know this is a lot of data, but the interesting thing here, I would say, is that with the dual site results, PMD inhibits contralateral M1 the most, in the sense that at 0 milliseconds, there's inhibition at 10 milliseconds, and there's inhibition at 50 milliseconds. And that's what all of this data shows here. So that's the takeaway point. The takeaway point is even more than the motor cortex, the premotor cortex is actually inhibiting and speaking to the opposite motor cortex. So she essentially, her second project is seeing, can we modulate that communication? And these are our different groups here. And essentially what she's found so far is that when we suppress the dorsal premotor cortex, there's an increase in M1 excitability. When we suppress the ventral, there's a decrease in M1 excitability on the opposite side. Unexpected, but kind of interesting, I think. So the clinical application, this is the last study that I'm going to be talking about. This is just the planned study, taking LNAS's results into subacute stroke, actually. Research has shown that increasing contralesional premotor cortex excitability can increase motor function, particularly for those who have severe motor functional impairment after stroke. As I showed, increased inhibition from the contralesional premotor cortex is associated with better motor function. That's what this shows here. As LNAS showed, the dorsal premotor cortex has quite strong connectivity with the contralateral M1. So dorsal premotor cortex is a viable target then after stroke. That's my conclusion. So we have individuals with subacute stroke recruited from the IURDPM within 1 to 3 months post-stroke. There's 3 intervention groups: suppression or theta burst, continuous theta burst stimulation to the dorsal premotor cortex. We're also enhancing or facilitating with intermittent theta burst stimulation, the contralesional premotor cortex, and then we have sham. And essentially what we're doing is, with all these visits, we're testing neuroplasticity, clinical motor function. We are priming motor learning with these different types of stimulation, and then we're having people come back afterwards to assess motor learning. So I'll end there and say that I hope that I've given you guys some interesting overview of the work that we've done using adjunct interventions like aerobic exercise and repetitive TMS, and how they can enhance neuroplasticity and motor learning and its potential applications in stroke. And I'll say thank you.